Alzheimer's disease is classified as a progressive disease, and it is the most common cause of dementia, which is memory loss and other cognitive abilities that interfere with daily life. It is the sixth leading cause of death and has no current cure. The greatest risk factor is increasing age. Most people with Alzheimer's are over the age of 65, but approximately 200,000 Americans under the age of 65 have young-onset Alzheimer's disease. What does Alzheimer's disease do? It impacts the prefrontal cortex and is extremely common for those with the disease to have difficulty remembering newly learned information. Some other symptoms are disorientation, mood and behavior changes, confusion, suspicions about family and caregivers, and can eventually lead to difficulty in speaking, swallowing, and walking. As brain damage spreads, cells lose their functionality and reach apoptosis. This causes irreversible brain damage. As mentioned previously, speech is affected in Alzheimer's patients. During speech, the higher frontal cortex responds by activating a speech motor control network, a distributed network encompassing primary and higher order auditory cortices and premotor cortex. My previous study illustrated that patients with Alzheimer's disease have an abnormally enhanced behavioral response to altered pitch in their auditory feedback, indicating abnormal sensor-motor integration processes in patients with Alzheimer's disease. In this study, I examined the neural correlates of the abnormal pitch perturbation response in Alzheimer's disease patients using MEG imaging. This type of imaging records magnetic fields produced by electrical currents occurring naturally in the brain. This study was performed by the participants phonating the vowel sound A, while a signal processor briefly perturbated the pitch of the auditory feedback. I then examined the high gamma band activity responses during the task. Now I will briefly mention the materials and methods that I used for this study. My subjects were 16 patients meeting the diagnostic criteria for Alzheimer's disease. I attached the criteria from the DSM-5 for diagnosing Alzheimer's disease. The DSM-5 refers to Alzheimer's disease as minor or major neurocognitive disorder. 14 of the 16 patients overlapped with 19 patients that participated in my previous behavioral experiment. These patients were recruited from the research cohorts at the University of California, San Francisco Memory and Aging Center. Also on this slide, I attached a picture of a healthy person's brain versus someone that has Alzheimer's so you can tell the difference. All of the participants in my study underwent a mini mental state examination. On this slide, there's an example of the types of question that might be asked on this examination. In a structured caregiver interview, clinical dementia rating scale and clinical dementia rating sum of boxes was determined for each patient. Set shifting was assessed by modified trail making test. The modified trail making test requires participants to draw lines linking items marked on paper and serially alternate between numbers and days of the week. This type of test is used to measure the person's ability to unconsciously shift attention from one task to another efficiently. Cognitive control was assessed by the Stroop test. The Stroop test requires the participant to read blocks of text where the name of the color of the text and the ink color of the text are the same, and then again where they are mismatched. This measures the participant's cognitive functioning, attention capacity, and skills and processing speed ability. Here I have attached a picture of the gamma activity recorded during the pitch perturbation response. This is shifts in pitch in their auditory feedback. This shows the activity for the controls and the Alzheimer's disease patients in their left and right hemispheres from 100 milliseconds to 300 milliseconds. The red regions show high activity levels and the blue regions show low activity levels. Here I have provided a breakdown of the gamma activity seen in the control patients for my experiment. During 100 to 200 milliseconds, the controls showed elevations of high gamma band activity over the left prefrontal and left posterior parietal cortices. They also showed high gamma activity in the right prefrontal and parietal cortices from 100 to 175 milliseconds. After the 200 millisecond mark, high gamma activity diminished. During the 200 to 300 millisecond, high gamma activity in the left anterior temporal lobe and the right posterior parietal cortex were significantly below the baseline. These results are consistent with strong involvement of higher order auditory regions, premotor, and frontal cortices in detection and correction for auditory feedback errors. I have also provided a breakdown of the gamma activity seen in the Alzheimer's disease patients. They showed a weakened response over the higher order auditory regions and virtually absent high gamma activity in the frontal cortex. During the 100 to 200 millisecond time frame, the patient showed a significantly high gamma activity over posterior parietal and occipital regions in both the left and the right hemispheres. They also showed high gamma activity in the left ventral motor cortex between 175 to 200 milliseconds. They showed reduced high gamma activity over the left superior precentral regions throughout the duration. The only consistent pattern in both the controls and the patients was the reduced left anterior temporal lobe activity from the 200 to 275 millisecond time frame. Next, I investigated the specific neural underpinnings of the elevated pitch perturbation response in Alzheimer's disease patients. I identified 8 regions of interest, and these regions were ones that showed statistically significant differences between the patients and the controls. The regions that I identified are shown in the figure on this slide. I wanted to find the specific contributions of the activity patterns of the 8 regions of interest. I used a generalized linear model with the peak behavior response and as the dependent variable and the activity patterns in each of the 8 brain regions and group identifiers as the predictor variables. This model explains 61.3% of the variance of the peak behavior and identified 2 key regions of interest as statistically significant predictors of peak behavior response. The 2 regions that were identified were the left prefrontal cortex and the right middle temporal cortex. Lower activity in the left prefrontal cortex predicted higher peak behavioral response, and higher activity in the right middle temporal region predicted higher peak behavior response. Part A shows the analysis of covariance model on the combined cohort of both patients and controls. The average high gamma activity in the left prefrontal cortex across the 100 to 250 millisecond was negatively correlated with the peak behavior response. Part 2 shows the R-squared of the model predictions and the p-value of the association between the left prefrontal activity and the peak behavioral response in each 25 millisecond window. Part C shows the average high gamma activity of the right posterior temporal cortex across the 200 to 300 millisecond post-perturbation onset. This was significantly positively correlated with the peak behavioral response. Part D shows the R-squared of the model predictions and the p-values of the association between the right middle temporal activity and the peak behavior response in each 25 millisecond window. The results from my study showed that Alzheimer's disease patients showed significantly reduced left prefrontal activity during the early phase of perturbation and increased right middle temporal activity during the later phase. Impaired prefrontal modulation of speech motor control network and additional recruitment of the right temporal regions are significant mediations of sensor-motor integration in patients with Alzheimer's disease. This demonstrates temporal evolution of speech motor control activity. These are contributors to abnormally enhanced pitch perturbation response. This impaired neural modulation signified the contribution of cortical network dysfunctions to cognitive and behavioral deficits in Alzheimer's disease patients. Pitch perturbation response is involuntary, which minimizes confounds. Abnormal pitch perturbation means that Alzheimer's disease patients have abnormalities in low-level sensor-motor integration processes. It is likely that left prefrontal cortices are essential for both speech motor control and executive function, both of which are disrupted in Alzheimer's disease patients. The results from my experiment could be useful to provide a scale to gauge disease progression of Alzheimer's disease patients. It could be useful to help treat patients in clinical trials, and could also be useful for the development of early intervention methods. Some critiques of my study are that the neural activity patterns contained both the auditory afferent and motor efferent processes. These are difficult to separate out. I also did not present analysis of age-related changes in sensor-motor integration of pitch perturbation of healthy young and old participants. My study only had healthy older subjects. Some future studies that I could perform would be to compare young and old healthy adults to understand age-related changes. This experiment could be performed again on a larger sample size. And I could also take this experiment a little further and see if population demographics affect the progression of Alzheimer's disease, such as maybe rural America versus urban America.